Drug development can feel like a black box. So today, Gail Cockwell, a biopharma expert with over 24 years of experience, is here to walk us through the whole journey. Gail is a pediatric rheumatologist who started her career at Pfizer, working across filings, launches, and approvals before moving to progressively smaller companies over time. One of Gail's real strengths is taking complex science and making it land for very different audiences, whether that's doctors, investors, lawyers, or patients, while keeping the core message consistent throughout. So let's start at the beginning and map out the full drug development journey, from that first spark of discovery all the way to a medicine reaching patients. It starts with a scientific discovery in a research lab. Then, chemists build a compound to act on that mechanism, which gets tested in animals for safety and efficacy before eventually moving toward human trials. Starting from a brand new biological pathway can take a decade or more before reaching humans, while building on a proven mechanism, where another drug has already shown success, can offer a much shorter path. Choosing an animal model comes down to two things that actually converge quite early: the science pointing to where the mechanism is most likely to work, and the real-world medical need showing where a new treatment would matter most. Before any human is ever involved, there is a whole world of chemical, toxicological, and pharmacological work happening. And that is what preclinical development is all about. To find a lead candidate, chemists evaluate how tightly a compound binds to its target, how long it survives in the body before breaking down, and how much other medications a patient takes might interfere with it. Some pathways are surprisingly easy to target chemically, while others remain stubbornly out of reach, with hundreds of researchers across multiple companies spending over a decade unable to develop a viable molecule for a well-understood pathway. Animal studies move through three layers: toxicology across 2 species to check safety, pharmacology studies to understand how the drug moves through the body, and then disease models like collagen-induced arthritis in mice to see if the drug actually works. Not all animal models are equally reliable. Infectious disease models tend to be highly predictive because the disease can be closely replicated in animals, while Alzheimer's models have historically struggled to forecast what will happen in human trials. AI and machine learning tools are changing preclinical work by speeding up candidate screening, surfacing new potential targets, and compressing what once took a decade of painstaking lab work into a much shorter time frame. Moving into human testing requires one big first step: pulling all that preclinical data together into a formal package for the FDA, known as an investigational new drug application. All the chemical, manufacturing, and animal data gets compiled into an IND and submitted to the FDA, sometimes after a pre-submission meeting, before the agency clears the sponsor to begin the first study in healthy volunteers. Phase 1 starts with a single ascending dose study, tiny doses given to small groups slowly escalating, then moves to a multiple ascending dose study over about a week, with biomarker data collected throughout to guide future dosing. For a migraine, side effects need to be minimal because the condition, while miserable, is not life-threatening. But for a late-stage cancer patient with months to live, a much more toxic drug can be entirely worth it. The benefit-risk framework is never one-size-fits-all. It shifts depending on how serious the disease is, what treatments already exist, and exactly who the drug is meant to help. Approval is not the end of the safety story. Post-market surveillance can uncover rare side effects, long latency risks like cancers that take decades to appear, and serious signals that may eventually lead to boxed warnings or even market withdrawal. When a drug is given to healthy people to prevent disease rather than treat it, the rules change. Regulators demand a higher safety bar because some of those people may never have gotten sick in the first place. Treating someone who is already ill means they accept more risk for relief. But giving a drug to a healthy person to prevent something they might never develop demands a much stricter safety standard from regulators. Phase 2 is where the drug meets real patients for the first time, and the goal is to answer 2 big questions at once. Does it actually work? And what is the right dose? Phase 2 is really built around 2 equally important goals: proof of concept, confirming the drug helps patients, and dose selection, finding the dose that best balances efficacy and safety across a range the FDA expects to see. A drug can fail phase 2 not because it does not work, but because the wrong dose was used, the wrong patients were enrolled, Or the study lacked a placebo group in a disease where placebo responses are common. Running a small Phase 2a study alone gives you less to work with than combining proof of concept and dose ranging into one larger study. The combined approach costs more upfront, but delivers richer data and reduces the risk of missing the optimal dose. Phase 3 is where everything gets bigger, longer, and more expensive. It is the large-scale validation stage designed to confirm what Phase 2 suggested before a company files for regulatory approval. Phase 2 sees more failures as drugs stumble on efficacy or design issues, while Phase 3 success rates climb to around 45 to 50% or higher, reflecting that only better characterized drugs with solid proof of concept make it that far. The FDA and EMA can see the same drug very differently. The FDA tends to allow off-label use with appropriate label guidance, while the EMA may flag it as an adverse event, and both agencies expect data relevant to their own populations. Manufacturing does not stay the same throughout development. It evolves from small bench-scale chemistry all the way to the commercial production lines needed to supply a global market. Manufacturing moves through 3 clear stages: benchtop synthesis during preclinical work, GMP-grade production once human trials begin, and by Phase 3, the final commercial formulation. Pill shape, color, release mechanism, and all must be locked in so results stay consistent and the company is ready to launch.